Hi, my name is Lily Frierson, and for my journal club presentation, I will be discussing the gut microbes of deep sea fish species and the diversity of these microbes. So, to get started, my bottom line up front. What is the importance of this? Well, there is an abundance of microbial life in the ocean, and especially in the deep sea. It's estimated that 75% of oceanic prokaryotic biomass resides in the deep sea, but as a region, it's highly underexplored because of historic inaccessibility and other barriers. So, the question of this study was to investigate and better assess the microbial diversity of the deep sea environment and what some adaptations these microbes might have to the deep sea environment and their unique characteristics. The finding of this study discovered a abundance of novel species. This study improved the understanding of microbial diversity in these ecosystems, and this study gave us insight into the distribution of genes relating to antibacterial resistance and also bioluminescence. So, the background of this field, this study, all of it, to begin, the deep sea is an extremely difficult place to survive in. There's no light, freezing temperatures, really low food availability. It's difficult to live here. So the organisms that do have a lot of pretty intense modifications. On the background of microbiology in the deep sea, in 1977, there was basically a revolution because hydrothermal vents were discovered by a remote operated vehicle. And this changed science a lot, a great amount, because we realized that on Earth, there are organisms that undergo primary production without the presence of light. So we discovered these chemolithotrophic bacteria that live on these vents. They have symbioses with a lot of invertebrates, and there are what was thought to be a deserted ecosystem actually has hotspots of biodiversity and life. And so, as a field, deep sea microbiology focuses a lot on hydrothermal vents and also cold seeps, um, and this research focuses a lot on the adaptation to these extreme conditions, microbial roles in geochemical cycling, and how these environments work, and sort of less on symbiotic relationships with macrofauna like fish. Another significant piece of background is that in 2016, the Global Ocean Sampling Expedition essentially sailed around the world for 2 years and sampled water from deep sea up to the photic zone to assess microbial diversity on a global scale. And this was major in our understanding of biodiversity of microbes in the ocean. And over 1,000 genomes of uncultivated lineages were harnessed through this experiment. And then also some background on understanding fish gut microbes. There have been a few studies done, but they primarily focus on shallow water fish of commercial interest, like cod and salmon. So, research done on gut microbes of deep sea fish is pretty lacking. So, why do we need this? There's a few different reasons. For one, there's a lot of potential for species discovery in these underexplored regions. Um, and the ocean is a very diverse place. 34 of our Earth's phyla live in the ocean. So, understanding the ecology of ecosystems, which is pivotal in natural history, can really be unfolded when you look deeper into the deep sea, especially in a time of anthropogenic stress when organisms can start to act wacky and even go extinct. Solidifying our understanding of all sorts of organisms is really critical. And then also, the deep sea plays a major role in our global ecology, in that it mediates the biological carbon pump, which essentially supplies organic matter to early parts of the food web in the ocean, which has terrestrial impacts too. So, understanding how these food webs work is very important to understanding how all food webs and all ecology operates on our planet. And then, in addition, what's really interesting is that, as we all know, microbes have been used throughout history to develop medical technology, but especially deep sea microbes have been shown to possess anti-cancer potential. They've been cultivated for enzymes which have been used in drug formation, compounds used in pain relief therapies. There's an abundance of biomedical applications that can be harnessed from discovering new microbial species in the deep sea. So, looking at our methods, to begin, Using a research vessel, these scientists collected samples from 1,000 meters deep in the North Atlantic Ocean, sampling areas in Scotland, Ireland, Iceland, and Canada in the Atlantic. Um, they isolated the gut contents and for DNA extraction using Powerfecal DNA isolation kit. Sorry, and they also used the OmniGene gut kit, and then metagenomic libraries were prepared using the Nextera XT kit. And then moving on from that, they used Illumina whole metagenome shotgun libraries. That was their primary method of this study. Sequences were cleaned, low quality reads were removed, they're trimmed, so they cleaned everything up. We know from our course so far that Illumina has limitations and benefits like the massive throughput and the low cost per base, but also shorter available read lengths, and assembling whole genomes can be difficult and also the short sequences can increase the error rate. But for this study specifically, shotgun was the best choice because the researchers are anticipating numerous novel genomes to be discovered. And then in addition, um, researchers were allowed to harness the full microbial composition of a sample without the need for culturing individual microbes, which can be difficult or impossible for deep sea species because it can be really hard to replicate the environmental conditions. And then also, the shotgun metagenomics libraries allow the researchers to screen the data for the presence of genes of interest, like we'll get to later, the antimicrobial or antibiotic resistance genes and bioluminescence related genes. And then in terms of program for their analysis, they used an abundance of different R packages, but here for taxonomic classification, they use Kraken 2 using only prokaryotic genome sequences, and then they use SuperFocus for a rudimentary sort of analysis of the different microbial functions. So it sort of can create subsystems and classify microbes based on what genes they have present, which is pretty interesting. And then they also developed some high quality metagenome assembled genomes, or MAGs. They were able to reconstruct 111 from the samples. In doing so, lets us compare deep sea species with known species. So they used MegaHit, MetaBat2, and CheckM, which was really impactful. And then to identify the antibiotic resistance genes, they used MegaR's database, and then also to assess the presence of bioluminescence genes, they use the computational biology system of hidden Markov models to basically look for different genes related to bioluminescence, but especially luciferase encoding LuxA gene. And then, as I said, their statistical analysis was performed in R, as well as data visualization and using the network analysis of microbes. They had a paired t-test they did through R. And as I said, they are able to display the microbial composition and different gene patterns using software through R, which is really cool. We'll get to see it in the figures. It looks really nice. So moving on to the results of the paper, the bread and butter here. The first figure that we're going to look at is really a summary of the major phyla, as it says, but it's really a focus on diversity and then the taxonomic distribution of the microbes found. So, fun part of this paper are these pretty pictures of these beautiful fish. So, on the top left, labeled A, we have a fangtooth, and then to the right of that, we've got a lizardfish. Below that is a species of deep sea shark. This is a Portuguese dogfish, and then to the right of that is a sculpin. So very charismatic animals right here. I think they're the cutest. And then below that, we can see a map of the sample sites. So as I said, we're in the Northern Atlantic Ocean. They're sampling your Ireland, Scotland, Iceland, and Canada. And then on the right of that, we can see a pie chart, which essentially breaks down the phyla of microbes present. So one thing that's also good to know when you're considering the impact of why there's so much diversity, there's not very much nutrient input in the deep sea. A lot of people have background knowledge of whale falls. So you see if a large animal falls down and floats all the way down to the deep sea, you get basically a hotspot. It's its own sort of micro ecosystem of all these animals that will surround it. So fish guts are a lot like that. I mean, in an environment where there's so little life, the fish gut microbiome has, as asserted by the authors, a really concentrated amount of bacterial community because the environment is so dilute that it's a really, really great place to live if you're a gut microbe looking for consistency. So, back to the figure. One interesting part of methods that they use is that to minimize potential for misclassification of the eukaryotic species, these these sequence reads only used prokaryotic genomes. And then in terms of the results, so we see the three largest groups that pull out of this pie chart are Proteobacteria, Actinobacteria, and Bacteroidota. So these were the dominant phyla found from the 32 species of deep sea fish sampled, and they comprised a mean relative abundance of 63% plus or minus about 4%. So these are the major players. And what's interesting is that another major phyla that's listed here is Firmicutes. So Firmicutes and Proteobacteria are both prominent in the microbiome of marine fish everywhere. They're really, really common organisms. But what's super interesting is that even though we see here these are identified to phyla, a majority of these um, samples were not able to be identified very far down the phylogenetic line. So they couldn't necessarily be classified to genus, definitely not species. So it shows us that even though, yes, the microbes in these fish are proteobacteria, they're not the same proteobacteria in terms of um, genetic similarity to ones that might live in a cod at the surface, right? So it shows, as the authors were trying to assert, that there's a uniqueness of these microbes. They're not the same as surface level microbes, which gives us insight into sort of one of the main hypotheses of the author, in that there's some unique characteristics that exist in these microbes, which is really exciting. But the main takeaway from this section of the paper is really just setting the stage of there is so much here, and we don't know exactly what it is, which is pretty exciting. And then moving on from that, figure two, we kind of shift in that the authors developed, they used shotgun metagenomic sequencing on the gut contents and were able to reconstruct 111 metagenome assembled genomes, or MAGs. So here in the upper figure, we see the diversity of these MAGs, which just sort of makes the scope of how much diversity there is present a little bit easier for us to digest as readers. And it creates a solid visual for us to see how many different mags fall into each phyla, as asserted on the right, and how far we've identified them down, because most of them are pretty difficult to identify because there are so many novel species that have never been seen before. So it's important to note that while these mags lack the purity of individually sequenced bacterial genomes, they do provide a lot of information about strains that are really difficult to isolate, sequence, and culture, which is important, especially when you're using gut content analysis. It's not always easy to have these super clean reads. So that's important. And this also lets us analyze the diversity and functional properties of individual microbes isolated from the environment and then compare them with surface species, which is a main focus of the paper, because we want to observe how these species are functionally different, and using these mags lets us visualize that. And also, what's really really just capturing how novel so many of these sequences are, only two could be identified to a species, and 52 could only be classified to genus. So it really, really captures, of 111, if we only have 54 that could be identified to genus, just how novel and exciting and new this information is. So again, as I said, using these mags can sort of help to break down what it is that we do not know and visualize it more meaningfully, because it can be sort of difficult to conceptualize the uniqueness of these specimens, but here we can definitely see just how novel they are. And then in the figure below that, these are genus clusters. So essentially, using the percentage of conserved proteins, each node is a metagenome assembled genome, and they're associated to each other by their relatedness in terms of conserved protein. So when we look at this figure, we can see that taxonomically similar mags can be isolated from different fish species caught in separate expeditions in different parts of the Atlantic Ocean, which highlights how these microbes have successfully adapted to life in the deep sea, where they're not just surviving in one place in a really small niche, they're actually surviving across the ocean basin, which shows how successful these adaptations have been, which again, one of the primary interests of the author is observing the characteristics that make these microbes successful in colonizing the fish gut. So it's important to see how these aren't just small scale successes, but really they operate on the Atlantic Ocean basin ecosystem as a whole, which is really rewarding. And then moving on, figure 3 is a visualization of the functional diversity. So again, one of the major goals of the author was understanding the unique characters of these microbes, and also a big part of the characteristics which makes them unique is their functionality. So here we have a visualization of this diversity. So mags here that were assembled were analyzed in their functional diversity using super focus computational software. And then the heat map shows the relative abundance of the different functional categories. And then I know it's kind of hard to see because it's so small, but on the left side, or I'm sorry, on the right side, we have the MAG IDs. And then at the top, we have different functions of genes, like different proteins and amino acid sort of different, a lot of different, like virulence factor. And then to the left of that, we see 3 characteristics of location, depth, and host. So basically, this is a really far reaching analysis of the location, the position of these organisms, their function, and then what they are. So, what we can take away from this figure is that the functionally dissimilar microbes are coexisting, which tells us that they can occupy these different niches, but also coexist in the same depth, habitat, organism. So, that's really important to understanding the ecology and the symbiosis of these organisms, and that it can kind of, they're not necessarily in competition. They can work together. And again, there's not really a clear functional clustering. So this lets us see that the heterogeneity in the functional capabilities across the different mags kind of points out that these microbes can have really dissimilar functional attributes. So again, there's diversity taxonomically, but there's also functional diversity, which is really interesting and has super profound impacts moving into our discussion. And then also, many of the mags that couldn't be closely matched to a reference genome are functionally distinct. So again, we have taxonomic diversity and also functional diversity. And lastly, what was added in as an interest of the author is that there aren't a lot of antibiotic resistance genes, which is interesting because pretty much every ecosystem on Earth has been impacted by humans and has an abundance of antibiotic resistance genes. But here, they were found in less than half the samples. And when analysis was restricted to genes only associated with acquired resistance, only 5 of the 47 intestinal samples showed antibiotic resistance genes. So this sort of indicates that there's a really, really low exposure to human impacts in the deep sea ecosystem. And then lastly, there was a surprisingly low amount of bioluminescence genes discovered, which has impacts because in marine science for deep sea animals, it's relatively well agreed upon that these bioluminescent microbes operate in these very, very critical symbioses that can be used by the fish in terms or for hunting, reproduction, communication. So seeing such low amounts of bioluminescence genes in these microbes kind of questions the extent to which these free living microbes really use bioluminescence to gain access to the intestine of larger deep sea fishes. So looking at our bottom line up front again, we see that, again, 75% of oceanic prokaryotic biomass is in the deep sea, so there's a lot that we need to know. The key question was looking at the diversity of deep sea microbes in fish guts, their unique characteristics, and also how they might be adapted. And they discovered an abundance of novel species, an improved understanding of microbial diversity, and also insight into the distribution of bioluminescence genes and antimicrobial resistance genes. For discussion, so new information. We know that we don't know, basically. We have a better sense of how little we know. So we've better assessed the diversity of microbes through this paper, and we have a better assessment of the rates of antibacterial resistance and the low levels of bioluminescence genes. And we also know that we have to keep sampling and keep working because there is so much novel information present here. Broader implications with this much biodiversity, there's that much more potential for biomedical applications through, as I said before, novel enzymes, metabolites, etc. And then we can also further that research by examining how these novel enzymes and other biological concepts are specifically engineered to surviving in the deep sea, which is really interesting. And we also know that there's a lot more to look at down here, which means there's a lot more we need to understand in order to adequately assess anthropogenic change in this ecosystem. So we still don't know a lot of the taxonomic classification, the biochemistry, the ecological role, and the symbiosis of these microbes because there's such an abundance of novel species. I want to take a second to point out some of the strengths and weaknesses of the paper. I felt like there was a strong suit of addressing taxonomic and functional diversity. There were a lot of novel insights provided. Numerous bioinformatics tools and statistical methods were utilized, and a lot of limitations were overcome because this is a really tough environment to sample. But in terms of weaknesses, there were definitely some geographic constraints in only looking at one region of one ocean on our planet, and also a pretty small sample size with only 47 samples. And also, because of the nature of these organisms, this paper had to rely a lot on bioinformatic tools rather than actual culturing observations. And also, I felt like a big direction of this paper was that they wanted to investigate the different characteristics and functional diversity aspects of what adaptations and evolutionary traits these microbes might have developed, but they didn't really explore it. And I would like to see that in the future. So, in conclusion, the deep sea continues to present an abundance of new species. Every day, there's more to learn, and there's a rich phylogenetic and functional diversity in these ecosystems. Microbes specifically have unique opportunities to explore deep sea ecology and uncover potential really beneficial applications to human life in terms of medicine and therapies. And further research is absolutely necessary, not only to explore resources, but also to more comprehensively assess the impacts of issues exacerbated by anthropogenic change like ocean warming if we want our planet to stay happy and healthy. And then these were some questions that I posed for discussion. Thank you, everyone, so much for listening, for watching. I really appreciate it. And lastly, here are my sources. Thank you.